Welcome back. In today's class, I am going to discuss polyembryony and apomixis. Here, what is polyembryony and what is apomixis? How the polyembryony is formed and how the apomixis is formed. The first one is polyembryony. Polyembryony. Generally, the seeds which are having single embryo. The seeds which are having the single embryo. In some plants they are having more than, some plants they are having more than one embryo. More than one embryo. One embryo is called polyembryony. Is called polyembryony. Polyembryony means the seeds which are having more than one embryo. More than one embryo, that is called polyembryony. Here the polyembryony is discovered by Anthony van Leeuwenhoek. The scientist Anthony van Leeuwenhoek, he discovered the polyembryony in 1719. The polyembryony is discovered in the citrus, first observed or the first discovered in citrus plant. Citrus plant. What is polyembryony? Poly means many, more than one. Here the seeds which are having more than one embryo, that is called polyembryony. And the first time Anthony van Leeuwenhoek in 1719, they observed the polyembryony in citrus seeds or citrus plants. Here the seeds which are having more than one embryo, and why the seeds they are having more than one embryo, or how the polyembryony takes place in a seed. What are the reasons? What are the reasons? What are the reasons are formation of polyembryony? Here the first one is formation of embryo. The formation of embryo from the embryo sac. Embryo sac other than egg. The formation of embryo from the embryo sac other than the egg. Here generally it is the structure of embryo sac, and the embryo sac at the chelazal region, they are having the 3 cells, and those cells are called antipodal cells. And at the center of this central cell, this is the central cell, and at the central cell there are 2 nuclei present. Those are called polar nuclei. And at the microphylla region, a large cell is present, that is egg cell. And at the adjacent of that egg cell, the 2 cells are present. Those cells are synergid cells. Here the formation of embryo. After the fertilization, the egg get fertilized from the male gamete. This egg it become embryo. Here the first reason is the formation of embryo other than the egg cell. Here except egg cell, the other cells are present. antipodal cells are there, central cell is there, and the synergid cells are there. These cells are going to be developed into embryo. This is the first reason. What is the first reason? The formation of embryo, other embryo from the embryo sac other than the egg cell. It may be the embryos, they are developed from the antipodal cells, or they may be developed from the central cell, or they may be developed from the synergid cells. Here, the most common cells, they are involved in the formation of embryo. Those are synergid cells. These synergid cells, they are involved in the formation of embryo. This is the first reason. And the second one is, activation of some sporophytic cells from the ovule. Activation of some sporophytic cells from the ovule. Here, the sporophytic cells means these are maternal cells. These are maternal sporophytic tissues. These are nothing but nucellus. Here, the activation of some nucellus cells of the ovule, they are involved in the formation of embryo. Here, the nucellus is diploid in condition. The diploid nucellus or the embryos which are developed from the nucellus. The embryos which are developed from the nucellus. The embryos which are developed from the nucellus, those are called adventive embryony. That is called adventive embryony. Adventive embryony, the embryos which are developed from the nucellus, that is called adventive embryony. This is the second reason of formation of polyembryony. Here the example is citrus and mango. Citrus and mango. In citrus and mango, the nucellus, they are developed into embryo. These are the two reasons of formation of polyembryony in a seed. And next one is apomixis. Apomixis means the production of seeds. Production of seeds without fertilization. The production of seeds without fertilization is called apomixis. The production of seeds without fertilization, that is called apomixis. And those seeds are called apomictic seeds. Those seeds are called apomictic seeds. Then, how the seeds they are formed without fertilization? Here, the apomixis is a type of asexual reproduction. Apomixis is a type of asexual reproduction, and here the sexual reproduction is absent in apomixis process. It means the apomixis, asexual reproduction, it mimics reproduction, it mimics means imitate the sexual reproduction. It imitate or it mimics the sexual reproduction. Apomixis is a type of asexual reproduction. It means the absence of sexual reproduction, the seeds are going to be fertilized. It means apomixis is a type of asexual reproduction. It means the asexual reproduction, it imitates or it mimics the sexual reproduction. Examples of apomixis are Asteraceae family and some grasses. These are the example of apomixis. And how the seeds or how the apomictic seeds are formed, or what are the reasons of formation of apomixis seeds. Here first the sexual reproduction. What are the difference between sexual reproduction and asexual reproduction in apomixis? In sexual reproduction, there are two important processes takes place. Those are meiosis, and the second one is fertilization. These are the two important processes takes place in a sexual reproduction. Here the meiosis means it is, the first one is meiosis. Meiosis means it is the reductional division in which the diploid sporophytic generation, it get reduced and formation of haploid gametophytic generation. In meiosis, by the help of meiosis process, the diploid sporophyte generation, it become haploid gametophyte generation. And the second one is fertilization. Process in which the haploid gametophytic generation to become diploid sporophytic generation. It means the haploid male and female gametes get fused and they are formed diploid zygote. Fertilization, the haploid gametophytic generation become diploid sporophytic generation. These are the two important processes takes place in a sexual reproduction. But here in asexual reproduction, these two processes are absent. These two processes are absent in asexual reproduction. Means by the help of asexual reproduction, means without meiosis and fertilization process, the apomictic seeds are formed. How the apomictic seeds are formed? The several methods are involved. I am going to discuss only the important methods. The first method is diplospory. It is the important method or a common method. Here, what is diplospory? Diplospory means here the megaspore mother cell. Megaspore mother cell is always diploid in condition, and that megaspore mother cell present in a ovule. And this megaspore mother cell, it form the embryo sac. And that embryo sac is formed from the megaspore mother cell, that is unreduced embryo sac is formed from the megaspore mother cell. How the megaspore form the embryo sac here without meiosis. Here without meiosis process. Without meiosis, the megaspore mother cell, it form the embryo sac that is unreduced. It means the embryo sac is also diploid in condition. The formation of unreduced embryo sac from the megaspore mother cell, that is called diplospory. Here the embryo sac, it has the different types of cells. Those are egg cell and other cells. Other cells means these are antipodal cells, central cell and synergid cell. These cells are without fertilization. Without fertilization, the egg become embryo, that is called parthenocarpy. Here the egg, it become embryo without fertilization, that is called parthenocarpy. It means the embryo sac, it produce the diploid egg, and that diploid embryo, it become embryo without fertilization, that is called parthenocarpy. And the other cells, it may be antipodal cells or the synergid cells or the central cell, they become embryo, and that is called apogamity. Apogamity. The first method is diplospory. Diplospory means megaspore mother cell is diploid in condition, and that diploid megaspore mother cell become unreduced embryo sac without meiosis, that is called diplospory. And the second method is apospory. The second method is apospory. Here the nucellus, they are diploid in condition, and these diploid nucellus, they are for the embryo sac. They are for embryo sac. They are from the unreduced, the nucellus cells, they are produced the unreduced embryo sac without fertilization, without meiosis process. Without meiosis process, the nucellus, they are produced the unreduced embryo sac. It means the embryo sac is also diploid in condition. That process is called apospory. What is apospory? The nucellus, they are formed the unreduced, the diploid nucellus, it formed the unreduced embryo sac without meiosis process is called apospory. And then the embryo sac, it has the other cells, those are egg cell and other cells. Without fertilization, the egg, it become embryo, that is called parthenocarpy, and the other cell, antipodal cells, central cell and synergid cell, they become embryo, that is called apogamity. These are the two common methods. They are from the apomixis or apomictic states. Here, what is the importance of apomixis, or what is the significance of apomixis? Here, the progenies means the offsprings. They are formed from the help of apomixis. They are helps to maintain the characteristics of parents. They are helps to maintain the characteristics of parents. These are the two special mechanisms that takes place in angiosperms and some gymnosperms. Thank you.